This is a podcast about new crops. You're going to love it. Join us on The Cutting Edge, a podcast in search of new crops for Wisconsin.
Welcome to another episode of The Cutting Edge. I'm Stefan Mirsky, Outreach Specialist for Emerging Crops with UW-Madison Extension. Today we're recording from the field. It is August 17th, and we're at the UW-Arlington Agricultural Research Station for the Hemp Field Day, showcasing grain, fiber, and CBD hemp plots. We're going to hear from Professor Shelby Ellison and a number of her students talk about the research they're conducting here at the Arlington Station. This is a lightning talk format with about 5 minutes for each speaker, followed by a few minutes of Q&A. We kick things off here, starting with Shelby Ellison. I hope you enjoy.
All right, so this is our grain and fiber variety trial here at UW. So what you'll see, so we just cut down the borders yesterday. I will tell you when you are walking out here, this is the most important thing, is there are many like 6-inch nubs here that are specifically meant to trip you. So please be very careful when you walk out here because there's a lot of trip hazards. But we have 3 different types of hemp out here. We have a fiber trial. So a couple quizzes about hemp. So hemp, cannabis sativa. So there are 3 different types of plants. There are male plants. Male plants mature earlier. I'll go and grab some. So there's Cheyanne, there's a male plant right there. So there are male plants. They typically will flower about 10 days on average earlier than female plants. The female plants need to be pollinated by a male plant to produce seed. So if you're growing for grain, you need male and female flowers to produce grain. That's dioecious, so that means 2 homes, male and female. Monoecious plants, like corn behind us here, they have male and female flowers on the same plants. So there are some monoecious types of hemp where you'll see male and females. There's also some where you're going to see separate male and female plants. You could see over there in the grain, the males die down. So there's a lot of males that are starting to die down in here. So we have a fiber trial, and then we also have what is called a dual trial. So that's going to be grown for the stalk, which is where the fiber comes from, and the grain at the top of the plant, only from the female plants. And then we have a grain only trial. Okay, so that's what's going on there. This is part of the Midwestern Hemp Cooperative variety trial. So we grow these at University of Illinois, Michigan State, Purdue University, and here at UW-Madison. We also have farmer collaborators across all 4 states that help us with those trials. Some of these people are here. Raise your hand if you're part of the trials. It's a couple of people here, so thank you very much to our farmers that help with that. Pretty much we're collecting data on what performs well in the upper Midwest. So we collect data on flowering time, we collect data on insect and pest prevalence, we collect data on sex ratio, we collect data on maturity, and then we will process the heads for grain and take grain yield, we'll take fiber yield, we'll ret the plants and process them into fiber. So here you can see this is kind of the output of the fiber is that it has 2 main parts. The inner part of the fiber, which is the hurd here. So it's kind of like, almost feels like it's wood chips essentially. And then the long bast fiber, which is what hemp is notoriously known for. So this is the outer part of the stalk. This is the bast fiber. Very long, strong fiber. Got myself my Patagonia workwear pants on, 45% hot hemp pants. It's typically a more durable material. It's a little bit tougher, but it's good for work. I have some other products here that are made out of hemp. We've got more processed. Right now, hemp, one of the things that people are really interested in is exploring construction, either from an insulation point or from a building material perspective, because again of that really long, strong fiber associated with it. I'm very happy to talk about all of these trials. I think it's better. You'll see there's tremendous variation, both within a plot and across the plots. You'll see there's things like this here. Got Futura 83. I mean, these were planted in late May. They're pretty darn big, okay? So this is an excellent fiber plant. It's gonna be pretty hard to go through with a combine to get grain off of that. I don't know what we're gonna do yet, but then you'll also see some short little plants that have big seed heads that are gonna be good for grain production only. So we're seeing a lot of variability.
All right, so hello everyone, I'm Cheyanne. I'm an undergrad in Shelby's lab. I've been working here for about a year now. I'm a 5th year at UW-Madison. My major is plant pathology and my minor is organic agriculture, specifically in the regenerative agriculture area. And so for the past 8 months, I have passionately been working on the Midwest hemp pest survey. If you guys look at your little sheet here, there's a QR code to my survey right at the bottom here. So, and this is for the growers of hemp. In this survey, we're looking for the pest prevalence and severity. So common pests such as thrips, a bunch of your different insects, and then also some of your common diseases as well. And then from that, we're also looking at the management strategies that you're using to try and combat weeds, insects, and diseases. And then also looking at what kind of research proposals you guys might be interested in the future when it comes to pest management or best preventative management strategies. So yeah, this survey, like I said, I've been working very passionately on it for the past 8 months. With this research, we're hoping to determine what pests are common in what areas, depending on the amount of results or people we get to take this survey. We can do a lot with this research. Right now we have very limited resources as to what kind of pest preventative management strategies growers are using. And so that's where we're hoping to fill in the gap of data that we don't have right now. With that, we are then hoping to move that data on to figuring out what are you guys using, what's working best in the field, and then where can we allocate different research and resources to better provide for the hemp growers of the Midwest. I would strongly advise all growers to take this survey, but I am biased in that. And yeah, I think if you're interested in learning more about pest preventative management strategies and such, it could potentially be very helpful. Again, the more respondents we have, the better the data will look and the more we can actually do with the data going forward. It is a 10-minute survey. I tried to make it as convenient as possible for everybody. So I made sure one of my main goals were easy to answer, easy to read questions. I try to keep out all of the big jargon. Just making it as easy as possible for you guys to be able to take this. And then on top of this survey, we have 5 $10 Quick Trip gift cards for the first 50 people who fully respond to the survey and follow the link at the end. There's about roughly 50 growers here today, so if you all take the survey, you might be able to get a Quick Trip gift card. I know everybody could use a Quick Trip gift card.
All right, so like I said, this is the Cultivar Check program, or very basically, this is a high cannabinoid hemp variety trial. Don't worry about the name. It stuck and stayed, and now it's just, that's what the project is called. But really what this is it's just a variety trial, okay? What we have done is we partnered with SARE, the Sustainable Agriculture and Research and Education, to evaluate varieties for performance. What we're trying to do is determine which varieties are going to be compliant, which are going to be uniform, and provide growers the best opportunity for success. We are looking at things like uniformity, overall performance, early, late, or middle maturity groups, and all of these things we are doing not only here, but across the Midwest with growers who potentially, like maybe you, to grow these varieties out for us. We send you the seed, we pay for the testing costs, and we provide that material free of charge. But what we have out here are 28 of the best potential varieties that we have evaluated over the last 4 years. As part of the program, we bring in genetics and we test them. After 2 years, if they're poor performers, they get cut for the program, and we bring in new material to evaluate. Can anybody tell me what the threshold for THC concentration is in? Yes.
Let's let somebody else answer first.
They make the rules.
0.3.
It's too low.
0.3. 0.3.
0.3 what? .3 what?
0.39.
Point, no, .3, .3 what?
Delta 9 THC.
Total THC.
Total THC. All these legislation rules keep changing. Anyway, so what we do though is we take each of these varieties, and at 3 weeks, 5 weeks, and 7 weeks, we take them and we submit samples for cannabinoid analysis at Rock River Labs in Watertown. That's not just us, that's every grower that we partner with. And so what we decide is at each of those time points, at a 95% confidence, which of those varieties or cultivars are going to be compliant. And then we try to make recommendations for growers as to which ones are suitable for the region, not only from a compliance perspective, but from an agronomic performance, right? So in a few minutes, we're gonna pass these around, we're gonna have everybody do their talks, and I'm gonna let you walk through these fields and answer any questions. You'll be able to touch, smell, just mainly those two things, to those plants. And really, I just want you to get a feel for what's out here. It's a little early in the flowering period, so a little bit later in September would probably be the best time to get out here and actually look at the differences among the flowering, but there is still going to be quite a bit of differences. All the varieties that we have out here are in the sheets that we provided, so if you find something that you like, make a note of it. Each of the plants are individually tagged with that name of that variety on there as well. Okay? So now that we have a little information about just kind of the holistic approach that we have in this field, just a little bit of information. We started these plants in the greenhouse on May 15th, and we transplanted them in the field on June 8th. Okay, we did. There was soybeans in this field previously, and we did no extra fertilization outside of the incorporation of that soybean residue. There was no herbicides that were applied. We cultivated the field, and we laid the black plastic. We transplanted the plants, and then I've done 2 passes with the rototiller since then to try to clean things up a little bit. And when the rototiller snapped last week and stopped working, the crew became our rototillers. So you can thank the undergrads for coming out here and sweating for a few days with me. All of these varieties have been checked, I guess, before they come into this program. We have data on cannabinoid performance before we bring them in. We have to make sure that they're commercially available as well. We don't want breeders to send the material in. It has to be a variety you can buy commercially, and then it has to be one that's going to be available for the future as well. All of these are commercially available, and all of these have been in the program for at least this year, some in the case of 2, 3, or 4 years, depending on that variety. All the results from those previous years are going to be available on our Emerging Crops website if you want to see that information. So before I pass this off, because this is really just giving you an overview of the variety trial itself, are there any questions you have for me about the data we collect? Yes?
Not so much about the data you collect, but how big were these transplants when you put them out in the field?
That's a great question. So we had about 3 weeks of growth on them. The plants themselves were only about 6 inches or so. What we used, deep cell culture trays, so they're very, very deep. They are designed to go straight from the seed into the ground, so there's no extra transplanting to prevent them from getting root bound. It was incredibly dry. It was like drilling into concrete with the augers to get these plants into the ground, and you're going to see the result of that with some of the lodging and some of the changes in how the plants grow to adapt to that. But there wasn't much we could really do. It's been so dry, and we didn't have irrigation out in the field at that time. We did 1 pass of overhead irrigation to save the trial, and we went from being, are we going to have plants in this field, to this, with 1 overhead splash at the right time. We had not had to irrigate this field or our fields at Arlington for our research trials like this in almost 25 years, 27 years, according to Dr. Erwin Goldman. So this was a very strange year in terms of how little moisture that we got, but the plants have adapted very, very well. This is a simple case of drip irrigation, should I have it? If you can afford it, and if you can do it, it's always great to have, even if you don't necessarily need to use it at that time. Okay? Yes?
I've got one. So maybe I missed it, but did you plant these later than the other trials, so you try to avoid pollen?
So we try to avoid any cross pollination from these plants, but there is always going to be some overlap there. Regardless of when we plant these, we all expect them to flower at the same time because they're photoperiod dependent. And despite, popular belief, we're even seeing that in some of the grain varieties too, where they are also acting more photoperiod than we thought initially, that they're not necessarily days to maturity in every case. So we tried to eliminate that, and typically grain and fiber varieties are going to flower earlier, but as you saw, there are some that are still shedding pollen right now. And so there's definitely going to be you'll see some seeds that were set in the earlier maturing ones right now, but we're hoping that the pollination period will end and we will actually be able to mitigate that seed production as we go through flowering. But outside of putting these at very, very far locations, this is the best we can do for a buffer. But great question. Yes?
Do these plants have the same some male plants, some female plants needed?
They can, but we hope that they don't when we buy them. So when we buy these seeds, we're told that they're feminized, meaning that we are expecting 99% of them, if not more, to be female only. So that's the expectation, and things have gotten better. But the reality is we still get males that show up. We get hermaphrodites, which are both male and female on the same plant, which show up. Seed certification is virtually non-existent, especially in high cannabinoid hemp. So what you see and what you're told isn't always what you're going to get. And that's why we have this project. So hopefully we can eliminate some of those issues. There will be plenty of time to walk through and ask me some questions if you have any, but I'm going to go ahead and pass this off to our next speaker, which is Sean Kim. Sean Kim is a master's student in the Ellison lab, and he's going to be talking about anthocyanin production in cannabis.
All right, sorry about that. So my research involves purple pigments in cannabis, so trying to understand the environmental stresses that cause the highest expression. I get asked a lot, why do we care about purple pigments in cannabis? If you're growing for floral production, so you're growing for the cannabinoids, it's a really popular trait. It helps drive the market a lot. And nowadays, for some reason, growers and breeders are making that a target that they're trying to reach only because it's not moving. If your cannabis is very green, they notice that it's not moving in the market as much as something that has a little bit of a purple hue to it. And they don't know whether the consumer is really educated on if that's better for them, if there's health reasons behind it or not, or if it's just aesthetically pleasing. But luckily, these pigments help out the plant as well a lot. They help against environmental stresses like nutrient deficiency, UV exposure, and then dropping temperatures. So once it's starting to get really cold out here, we start to notice that some of the plants will turn purple. But it's not all cannabis plants that have the ability to turn purple. There's a genetic component behind it as well. So we're trying to figure out if the plant can be purple or not, and if it can, how can we get the highest expression out of it. So my research was mainly with temperature treatments. We're looking at mainly controlled environments. We wanted to take UV out of the equation and see if we drop the temperature, what's going to happen to the purple pigments in cannabis. So we selected a variety that turns purple every single time. It's just dependent on its age, and then put those in different cold treatments, starting with almost freezing up until room temperature. And we noticed that there is a sweet spot where the plants will turn very dark purple, and we're trying to understand if there's a trade-off for cannabinoid content and whether or not that if you drive that secondary metabolite in the anthocyanin direction, if you do have a trade-off for cannabinoids. So we're looking into that right now. And then as well, we created a mapping population. So what we're doing with that is essentially trying to figure out the genetic component behind this. So we took a green father and then a purple mother and crossed those two. So the progeny should have about 50% purple, 50% green. And we do a back cross to the green father, and then continue that on to try and narrow down that little bit of the genetic component that's causing the plant to turn purple. And then we can start analyzing that data later on. So that's where we're at right now. I've been collecting DNA from all these plants in the field last year and then on our populations. And then with the help of some postdocs, we're going to try and analyze that data and see if we can find some significant QTLs. So that's my main research and what I've been looking at. We also looked at UV exposure as well. You'll notice that some plants that are facing the sun will turn purple on one side, and then if they're in the shade, they won't be as purple as much. And also we noticed tags around the stem. Sometimes it's purple stems all the way down, and then wherever that tag was, you'll see that it's green. So we've just been taking note on all this to try and see how we can get the highest expression for our plants that could potentially lead to a more purple flower for growers. All right.
Okay, hi everyone. So I don't have a slide to guide me, so pardon me if I just swing around. My research work with the Ellison lab is mainly around collection, characterization, and preservation of feral hemp, feral US germplasm. And as we all, you might not know, but up till now, there had not been a national repository for hemp collection in the US. So there's a new one created by USDA at Connolly in Geneva. And so part of my work is to make collections around the US, characterize these materials, and then try to populate the gene banks. Now, how do we do this? So my job is to go on iNaturalist, which is a social app where you have a lot of citizen scientists, and I think some people here have. So on iNaturalist we have citizen scientists, we have researchers, and every kind of people interested in all sorts of things, animals, plants, all sorts, all species types. And then what they do is when they find maybe feral hemp or ditchweed, as some people call them, they take a picture of that and then georeference it and then upload it to the iNaturalist app. So my job is to go to the app and then pull up all of these and then try to go to those locations to see if what were really reported are feral populations or ditchweed, or maybe someone just planted hemp behind their houses and then they think this is feral or something. So I go there, I make the collections, and then so far I've made about 1,050 collections across 8 different states in the Midwest, and most of all subset of all of these have been characterized using like genotypic markers that tells us if these are CBD types, if these are the drug type cannabis, and if they are intermediate, that is just in between. So and then we also go further to use the HPLC analysis just to be sure that what we are dealing with is not the drug types, to further screen. And then when we find the drug types, of course we send that to our collaborators who they have a way of getting rid of those, because we are not interested in the drug type for now. So so why are we doing this? The whole essence of this is to to be able to have as we all know, genetic diversity is like the background for breeding, no matter the kind of crop. So the essence of this is to to have this genetic diversity where breeders can go in and just go to the germplasm and request for what they want, and then they can head out to the field and just make crosses of superior feral materials. So for our project, I don't want to forget this. For our project, if you think you have feral ditchweed behind your house, you can contact us. And then if you also can go, if you're hikers or something or conservationists and you go around and you know where these populations are, you can make collections for us, and we have like $150 to $200 per unique population for each collections that you make for us. So all right, that's fine. So if you let me know, and then I can send you a kit, and then it also comes with a license for collection, and then the protocol, and then just some writing materials and all of that. It's not of a big deal. You just chop up the inflorescence, the seed bearing parts, dry them, room temperature, and then just bag them and then just label them. So it's not a whole process, because I mentioned protocol, you might think it's something difficult. So that's that. And then out here we have some of those. Those unique populations have to be like 5 miles apart, so we are sure they are kind of unique and not just out crosses. So out here we have some of those materials planted in the field, and if you take a look, you see the very tall plants out there. Those are the feral types that are being characterized for something like flowering dates, taking flowering notes on those, and then checking their sex to know if they are male or females. And then just mostly what we are looking at is just the flowering, taking flowering notes on this, because of course it's very important. When you're doing breeding, you want the plants to be about the same maturity period, so you can make all crosses at the same time. So we're just checking that out to see. And I think that's all for now, but if you have questions, I can take that. But before I forget, I have a, can you get this for me? No. Yeah, so can just, that's, I've gotten questions from the first group of people, what a male hemp plant look like. If you take a look at that closely, you see it has like 1 million pollen sacs there. So that's the male plant. They usually mature early, and of course they die faster. And for the female types, they're usually, the flowers are shaped like the teardrop. You have a ball, and then you have like a needle-like stuff just sticking out. So that's the difference between the male and the female. And then some of them are monoecious. They have both the male and then the female. All right, so I'll be happy to take your questions later in the field.
Okay, now that I'm all buckled in. Yeah, so my research was in companion cropping and cannabis. So companion cropping is an indigenous cropping system. So basically it's the idea that you have a couple of different crops in the field, right? And they're all giving each other something that they need. So it started with the 3 sisters, if you're familiar with that. So maize, bean, and squash, each of those giving the other something that it's needing, right? That usually in a monocrop system we have an outside input for, but I really wanted to see how we could have a cannabis best production practice that didn't really need a lot of outside inputs. So I wish that we were at a field right now with some companion cropping, but I can lay the scene out for you. So basically my project looked like this, but then in between each of the rows was a companion crop that we planted. So where you see bare soil, there was a crop there. So we had 5 different companion crops. We did basil, marigold, dill, cilantro, and sage. And then we also had a blink treatment, which was to replicate a monocropping system. And I looked at a lot of things. So I had 2 objectives that I kind of focused on. So one was companion cropping and the effect that it has on cannabis yield. And the other was the effect that companion cropping has on the ecology of the land that we're planting on. So for the first objective, I did normal things like plant height of our hemp, and then I also did biomass, so weighing it, and then cannabinoids, so THC, CBD, things like that. And then for the second objective, I really looked at insects. I did microbiome work, and then I harvested our companion crops to see what kind of additive income they could have for growers. And yeah, I really kind of tried to look at ecosystem services. So ecosystem services I think of as things that the environment gives us and things the environment gives itself. So the only reason we're all here today is because of ecosystem services, right? We are able to breathe air because of them. And so for this project specifically, I was really interested in things like weed control and erosion control, things that a healthy environment should be able to do for itself. So to get into some results, because I have a lot of those, because I just kept adding things, and when you add things, you get lots of results. So we saw that companion cropping didn't have any effect on our hemp yield. So the things like height, biomass, cannabinoid content, which is really exciting. So it means that really any of the companions that did well, a grower could plant, and they could be pretty certain that it wasn't going to affect any kind of yield at the end of the season. Any sort of genetic or any sort of variability in the field still with cannabis is typically genetic, so it'd be interesting to see once the genetics are a little more sound if companion cropping does have an impact, but for right now it didn't. And then I saw that our companion crops like marigold and basil did a really good job at weed cover. These are really early establishing crops, so they were able to really like fan out within the rows before the hemp got big and was able to outshade a lot of weeds. Those crops were able to do that for them. I surveyed that by a weed survey, so weed percent cover. So I did like quadrats in the field, and then I assessed what was in there by percentage. So I'd be like, in this 1 by 1 foot quadrat, there's 60% basil and 40% or 30% weeds and 10% ground, right? So things like that to kind of understand how our companion crops are competing with weeds. And then for things like insects, I did sweeps in the field at 2 different time points, one at the beginning and one at the end of the growing season. And then I gave those to entomology students because I'm not an entomologist. I can't characterize insects or assign taxonomy.
So those were all identified for me. And then I worked with someone in entomology and gave them like a pest, beneficial, or incidental classification. Then I was able to understand what sorts of insects were around the different companions. So we saw that basil had more pest insects than our others statistically, which I really think highlights the importance of diversity in a field. So if we had just planted basil in between all these rows of hemp, we may have seen a bit of pest pressure on the crops that we were planting. But because there were a lot of different crops like dispersed within our field, we didn't really see a ton of pest pressure or pest damage on the hemp or the companions, which was really exciting. Yeah, I could go on and on and on about companion cropping, but I can have some questions. Yeah.
The plants that you picked are mostly herbs. How did you pick those plants? What plants did you consider and reject?
Yeah, so this I inherited this grant. It was already funded, so I didn't get to have the I wasn't able to help with the selection, but mostly these crops were chosen just because of either them being used by home gardeners or them being used by indigenous peoples. So we kind of went off of that. There's not really a lot of literature with companion cropping right now, like scientific literature. So we really went off of what has been good for other people. Yeah.
You said you looked at the economic benefit of those
middle
crops for those things. What is it for marigolds?
Oh, see, yeah, I completely forgot to follow up with that. There's so many things. So I harvested half of all the companions and I weighed them and then put them in a little formula that I made and did a farmer's market value. So marigolds did pretty well. Yeah, basil definitely did the best because I was able to harvest it multiple times. And then on the same level were things like cilantro, dill, and sage, or not sage, cilantro, dill, and marigold. Yeah, the one you were asking me about. And then underneath that was sage and blank. So yeah, but they did pretty well, definitely.
I didn't realize people bought marigolds.
Yeah, they do. A lot of the times, I think they would do well in if there were more flowers also grown, because they do well in bouquets. Yeah, I don't think people typically just buy marigolds, but they add a really nice color into some bouquets. Yeah. Yeah, and I'm happy to answer any questions in the field as well.
You're not going to make me start over, are you?
You don't have to.
Good. I'm with the Integrated Pest Management Program here at the University of Wisconsin. I am the state liaison for the IR4 program, as well as one of 2 field research directors for the IR4 program. You're probably thinking to yourself, what in the heck is the IR4 program, right? Yeah, okay. It merely stands for Interregional Project Number 4 of the USDA. Why should you care about it? Many of you are aware that we now have a herbicide registered on hemp, right? Recent registration this past year. That is completely the efforts of IR4 to get that registration in place. That was begun in 2020, was a very short turnaround, resulted in us getting this registration here in 2023. So IR4 is a program, a national program, that helps to secure pest management registrations on minor use crops. Okay, those would be crops on which uses are less than 300,000 acres across the United States, or it can be a minor use on a major crop. So we will occasionally do something on field corn or rice or wheat or alfalfa or something, but it's a very minor use, not something that's going to be used by the vast majority of growers. Okay, so we help to get those registrations in place. Why is it needed? If you're a registrant of one of these pest management products, some of the numbers that get thrown around are it takes about $350 to $500 million to bring a new active ingredient to market. Okay, that's a lot of money, right? They have about a 17-year patent life, right, on that product when they bring it to market. Probably 6, 7, 8 years of that patent life are going to be used up in the research and development phase of getting that product to market. So now they've got 10 years or less of trying to recoup that major investment. To be perfectly honest, they have no interest in registering or doing the data and the work that it's going to take to get that registration on hemp. Or in Wisconsin here, a whole litany of crops. I mean, onions and carrots and mint and peas and cabbage and sweet corn and snap beans. The list goes on and on. So that's where we come in. We do the work, collect the data that's needed, and produce the petitions to EPA to get the tolerance for those pesticides to be able to be used on that crop. Okay, so what does that look like? Behind me here, I've actually got, I guess, 4 different hemp trials out here this year. The one behind me here happens to be for abamectin, the active ingredient, a miticide. It's purely two little plots. This is a treated plot here. That's an untreated plot in the back. The use pattern that's being proposed is 3 applications at a 7-day interval. So right now I'm in the middle of making the applications on this front plot here. I think I just made the 2nd application yesterday. I'll be making the next application in 6 days, and then the pre-harvest interval will be 7 days after that. I'll come out, I'll harvest this for seed, I'll harvest it for fiber. Those samples will get frozen at -10, taken by semi at -10 degrees Fahrenheit to a single lab in the United States that's going to be analyzing for the pesticide residues on that crop. This trial will be repeated, or is being repeated, at 10 other locations around the U.S. as well. The exact same protocol, we're all doing the exact same things. So instead of replicating it here in Wisconsin, we're basically replicating it around the country in different growing environments and in different conditions. So once that data all gets put together at headquarters, they will then use that to petition EPA for that tolerance. Now, I wish I could tell you this is a nice, quick, and easy process. For the ethylfluralin or sonolin herbicide that was recently registered, it was. It only took us about 2.5 years. The reason being, they were able to do that with no data. Okay, they used Canadian data, no U.S.-generated data, because Canada had already done all of the work, and we do cross lines quite a bit with our data. We share with Canada, they share with us. So we were able to take that data. We were able to take the knowledge that any of the residue work we've done with ethylfluralin, it has never shown a residue in any crop because it's not translocated in the plant. And we were able to extrapolate that to hemp, take that to EPA, and they agreed. They said, yes, there's not an issue here. We can go ahead and go with your proposed use pattern and tolerance and provide that. Now, we were very fortunate because that's only the first step in getting the registration. Just because EPA says it's okay and publishes that tolerance, it's not acceptable to use that product or legal to use that product until it shows up on a label. And the registrant doesn't just automatically say, oh yeah, we're going to add this to the label, because every time they open up that label, there's a cost associated with that. It has to be reviewed by EPA, and it opens them up to any of their existing uses on that label being looked at by EPA as well. So it could require multiple modifications in the end. So sometimes what they'll do is they'll say, you know, we've got a couple of other uses we're looking to add in about a year and a half down the road. We're going to sit on this one, so we only have to open the label once. We were fortunate. This tolerance came through. The registrant quickly opened up that label and added the use, so we got that going forward. In a normal case, we're looking at a much longer process. So this field process generally will take about 2 growing seasons or 2 years to get all of those trials into the lab, and then an additional year at the lab to get all of those samples run and get the data put together. IR4's timeline that they are trying to stick for is about 36 months from the time they initiate a field trial until they have the data put together and ready to submit to EPA. Once it gets submitted to EPA, it just gets put into their work plan, and that takes a while too. Quite honestly, it will often take up to 2 years before it gets through the work plan at EPA. So now we're out to 5 years, maybe another year before it gets added to a label by the registrant. So we're looking at somewhere between about a 6 to 7 year process from the time I'm doing this trial here on abamectin until you're actually probably going to see a label. But to be honest, it's the only thing we have going. I mean, you're never going to see it being done by one of the registrants. So it may not be the fastest process in the world, but we're guaranteeing that we're producing good data by the way we're doing it around the country, and that we're getting the things that need to be done for EPA to approve it. So what does it take as far as these trials? You'll see everything I've got here is on grain and fiber. Hemp being so new, EPA really was kind of struggling. They didn't know. They actually asked us to do one residue trial on hemp because they didn't know what even plant parts they needed to analyze to see where the residues might show up. So in the end, unlike many other crops where they say, okay, one trial has to be done in region 5, which is an upper Midwest here. One has to be done in region 12, which is the Pacific Northwest, and very specific so they get spread out. With hemp, they actually accept any 8 trials across the United States. So IR4 will tend to do about 10 of them. But of those 8, 5 of them have to be fiber and seed trials, and 3 of them have to be CBD trials. Okay, so this same protocol is being done on CBD as well, because right now they don't have a mechanism to differentiate between the two. They're looking at registrations that go across both CBD and grain fiber. In addition, one of the trials has to be what's called a decline trial, in which samples get harvested every couple of days so that they can produce a curve in the data as to what that residue looks like. Another one of those 8 trials across the U.S. has to be what's called the processing trial, in which the seed is harvested, a larger quantity. In fact, actually 2 of mine up here will all have to harvest 10 or 12 pounds. It'll get sent off and it'll be processed into flour, into hemp hearts, meal, oil, everything you can think of, and then they will look for the residues in those final processed commodities. Okay, so what sort of things are in process right now? As you know, the Sonalan or ethylfluoralin is the only currently registered product. There's one other herbicide that's being looked at right now, and IR4 has trials in. I have one of them out here, and that's Quizalofop or Assure 2. It's a graminicide, controls grasses post-emergent. The only thing that's going to be a little tricky is the registrant has already warned us they will not accept an over-the-top application of it. They are concerned with it, so it's primarily going to be for between rows in CBD production. Okay. We have 3 insecticide trials that are being looked at right now. One of them out here is a fenpyroxamate, and that's for spider mites. One on cycloniliprole, and that's primarily on lepidopterans and thrips and the like. And then we have one trial out here, the one behind me, on abamectin, and that is also for control of mites. And then there are 3 fungicide trials. One on isofetamide, that's primarily for things like botrytis, powdery mildew. Another one on flutionil, and that's also for powdery mildew. And then finally, one on cyazofimid, and that would be more for pythium and some of those early diseases. So there are a number of trials in the hopper, a lot of things being looked at. Again, of these 7 that I just listed off here, I would say it's going to be at least another 5+ years before we see registrations on any of those. So hopefully that was a little overview of what IR4 is, why it's important to hemp production as this commodity grows. If anybody has any questions, I'm happy to entertain them. Yeah.
The 10 other locations, or the 10 total locations under this IR4 program, are they kind of up in this region, or they're just across the U.S.?
They are across the U.S. So there are probably, I'd have to look exactly, but I'd say we have about 80 to 90 cooperators across the U.S. that do these trials, because as simple as this looks, this little block of hemp here requires me to do 200 pages of documentation in a binder. Okay, so I actually put my time at about 40 hours per every one of these trials, and I'm doing 25 of these trials this year. So it's an incredible amount of work, and in order to do it up to what's called GLP, or Good Laboratory Practice Standards, to meet EPA requirements. So, but yeah, this particular trial I know is being done in California. It's being done in Washington, North Carolina, Florida, somewhere in the Northeast. I'm not sure where as well. Yes.
What sort of pesticides are you trialing for the fiber usage, and what are the fiber processes that you're testing?
Yeah, so believe it or not, the fiber processes or processing, they are not actually processing the fiber to look at it because it's a non-food use. So therefore the fiber portion, even though it has to be, it still is required as part of the registration packet, because it's a non-food use, they're not actually processing fiber to look for residues. Okay.
And don't you wear fiber against your skin?
Well,
It's the largest organ.
yeah. Very true. Very true. But I yeah, no, absolutely. I'm just sharing you. We're going on EPA guidance, and based on their risk analysis, that's how they go about it. What trials? Again, all of these ones that I listed off here, they're being done on both CBD as well as grain and fiber. So it's across the board. They're not looking on individually on one or the other.
You don't have any special insect or fungus that makes the fiber yet less useful?
I'll be totally honest. I am not a expert in that area. I couldn't tell you. Yeah. Back here.
You mentioned that the Assure 2 was not ever going to be approved for over-the-top application for CBD. Do you think that would be approved at some point for fiber and for fiber?
No. Unfortunately, I've gone round and round with the registrant because I'm like, you're making this label useless. Don't waste our time. And yeah.
What's the lot? I mean, are they worried about it like casting off into other areas?
They're worried about crop injury. Okay, now we have a broadleaf plant. It's not going to affect a broadleaf plant. It only affects grasses because this does not have the site within the plant to affect it. It can cause some initial injury. The registrant's concern at this point, as with all minor crops, we don't know exactly what the value of the crop is. We don't know how, are people going to come after us with lawsuits if they see injury? Once they get more comfortable with a crop, they will start to open up a bit more. But initially, that just is kind of the blanket response. No, we're going the safe route. So.
Got 2 more questions.
Real quick. Yep.
What's the current status of seed treatment?
Of seed treatments? Yeah, right now we're not working on any seed treatments at this point. If there are seed treatments or a need for seed treatments, and this would go with anything, if there's a particular need within hemp that is not being addressed right now, that's really important to let Shelby know, to let myself know, because we are the people that at the national can submit those proposals. But generally we have to have some data. We have to have some efficacy data to show that it's there and that it works. But it's really important to let us know.
Damping off and lithium being one of the major.
Right. So right now the thiazofimid, not as a seed treatment, is the one that they're looking at. But they're looking at it both on CBD as far as on transplants, as well as they are looking at seeded as like a banded application over the seed. But to do a seed treatment trial adds a whole another level of complexity. So. Okay.
Well, thanks for listening, and be sure to check out the Emerging Crops website for more information on how to get involved in the different projects that were mentioned today. That website is www.emergingcropswi.org. Hope to see you in person at next year's Hemp Field Day.
Brought to you by the University of Wisconsin-Madison Division of Extension.